Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing ICMR BRET question paper. This is section B that I am discussing. This is cross-disciplinary section and this was conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of ICMR being discussed, you can go to my channel Omni Bioentrance. In the channel playlist you see for ICMR and you will find them there. So coming to the first question, a short microtubular organelle projecting into extracellular medium used for locomotion. So locomotion means for movement, and you can get confused with pseudopodium as well as cilium. Now pseudopodium, it is not a microtubular organelle, so naturally cilium will be the answer. But I will explain 1 minute. Cisternae, you all know they are flattened sacs or vesicles and they are found with the Golgi or a part of the Golgi complex. So that cannot be the answer. They are not used in any locomotion. Then coming to basal body, it is the structure at the base of the cilium or the flagellum, and usually for anchorage of cilia or flagella. That is why you used basal body. Then a pseudopodium, it is only a temporary extension of the cytoplasm, and it can be used for movement as well as for engulfing particles, and it is found in mainly in the amoeboid cells. So here it is cilium, that is the most right answer. It is a short microtubular organelle and it is projecting into the extracellular medium. Okay. Then organelle that is involved in carbohydrate digestion, autophagy, hydrolysis. Okay, here it is the only organelle that is mainly involved in digestion, autophagy, hydrolysis is nothing but your lysosome. They are suicidal bags and they have acid hydrolases, that is enzymes present in them. Then Golgi bodies, you all know they are involved in protein modification as well as protein sorting. Then nucleus, it contains the genetic materials and that is your DNA, and it regulates various cellular activities, and mitochondria is the powerhouse, that is it produces the energy through respiration. Then next one, microtubular protein having ATPase activity required for movement. If you know the definition of these options, then happily you can go for the answer. Now ecdysone, it is nothing to do with microtubular protein. It is a hormone and it is involved in insect development and molting. So if you know what is ecdysone, you can cross the answer. It is not the right answer. Then fibronectin, that is found mainly in the extracellular matrix and it is involved in cell adhesion, wound repair, tissue repair, etc. Now sarcopene, I don't know what it is. Whether such a term exists also, I am not sure. Okay, then dynein, it is involved in the transport of organelles and cargoes. Then during cell division, it is involved in the segregation of the chromosomes. Then it is involved in the movement of cilia and flagella. And another point about dynein is that it is involved in retrograde transport. That is, if you are taking the case of the neurons, it will help in the movement of this cargo and organelles towards the cell body. Okay, that is retrograde transport. And if it is in other cells, then it will move along the microtubule in the direction of the microtubule minus end. So that is what dynein is involved, and it is having ATPase activity. So the answer here is option 3. Then next one, which one of the following principle of bioethics emphasize the importance of minimizing harm and maximizing benefits in healthcare decisions? Now there are some principles in regards to bioethics, and all these, that is justice, beneficence, autonomy, maleficence, etc. All these come under these principles of bioethics. So justice means fairness or equity in healthcare access. Okay, that is healthcare resources all should be distributed or given fairly to patients or individuals or to community. Okay, that is what it means. That is, everyone receives a fair treatment and has equal opportunity for healthcare and research, and there is no discrimination between the patients who are coming to healthcare personnels. That is, the healthcare personnel don't discriminate the patients based on factors like race, religion or social status. That is what is meant by justice. Now what is autonomy? Autonomy, it refers to the patient's right to make decisions regarding their own healthcare. Now sometimes the patient may take unwise decisions. Okay, so even if the patient chooses or makes unwise decision, the healthcare personnel cannot object the patient from taking this decision. That is, if the patient chooses to go to hospital B instead of going to hospital A, just because the patient is not given information that hospital A is having more facility for his for the treatment of the disease from he is suffering than hospital B. So the patient can choose the decision, okay, I will go for hospital B instead of hospital A, just because he is not given information regarding the facility in hospital A. So what the healthcare personnel has to do here is, he has to disclose all the information to the patient so that the patient can make an informed decision about his healthcare. See, the patient going to the healthcare personnel is not always informed about what is happening to him or what is his health condition, what are the risks he will face in future in his life if he doesn't take proper healthcare decisions. He doesn't know. So the patient should be given full guidance regarding his health care decision by the healthcare personnel. Then the healthcare personnel shouldn't pressurize the patient and influence him by telling him, yeah, you go to that hospital instead of this hospital. The healthcare personnel doesn't have the right to tell the patient. He can only tell, see, this hospital will give you better facilities than the other hospital. Now the decision is upon the patient whether he has to, which hospital he has to choose for his health care. Okay. Then the patient who is taking this decision regarding his health care must be mentally sound, and he should understand the information that the healthcare personnel is giving. That is called as autonomy. Then beneficence means the healthcare personnel has to act for the benefit of the patient. And non-maleficence means to avoid harm to the patient. That is, harm means the healthcare personnel shouldn't give any physical or emotional or psychological harm to the patient. That is what is meant by non-maleficence. Now here in the question they have told minimizing harm and maximizing benefits. So as per me, both 3 and 4 are correct. Beneficence means the patient is getting maximum benefits. So there is naturally minimizing harm there. Okay, only if you minimize harm, the patient will be getting maximum benefits. Now non-maleficence means minimizing harm. So maximum benefit for the patient. So both these terms, that is beneficence and non-maleficence, they are intertwined. Okay, so both these terms, they both are correct as per this question. So, but your answer key, they have given non-maleficence as the answer. Now next one. Which of the following ethical principles support the idea that healthcare resources should be distributed fairly among individuals and communities? I just told now it is justice. Okay, just now we discussed in the just previous question. So the answer here is justice. Now what is this confidentiality? Confidentiality means the personal information regarding the patient is kept private and secure, and you have the healthcare personnel has to respect the patient's right to control who has access to the medical details. Okay, that is what is meant by confidentiality regarding the health condition of that particular patient. Then informed consent, that is a legal requirement, and the patient have to should be given relevant information about the purpose of this particular medical treatment, so that he can either accept the treatment or he can refuse it. So that means the healthcare personnel, they have to give sufficient information regarding the risk, regarding the benefits, and if there are other alternative for the treatment, that the patient has also got the right to ask questions to the healthcare personnel, and that is called as informed consent. And only then the healthcare personnel can further further take the treatment to higher levels. Then next one, these are the glands and the hormones that are released from them. So the first one will be released by anterior pituitary and affects adrenal gland. So A will be ACTH, A4. A4 is there in 2 and 4, so we will not see 1 and 3 are not correct. Then next one, influence extracellular fluid volume, that is your ADH. So B3, okay, 2 they have given B1, so that means option 4 is the right answer. Okay, we will see the others. Stimulate estrogen secretion and egg maturation, okay, follicle stimulating hormone, so C1. And causes uterine contraction during childbirth is your oxytocin, that is D2. So A answer is 4. Then next one, given below are two statements. Choose the correct answer from the option. Okay, we have to say which statement is correct, which is wrong, or both are correct or both are wrong. Okay, now see the first statement. An endocrine gland releases substances through tubes or duct. That statement itself is wrong. If you remember, endocrine glands are ductless glands. So where will they release? So this statement telling that they are releasing substances through tubes and ducts is entirely wrong. And endocrine gland, they secrete what is called as hormones, and they are released directly into the bloodstream to be transported to different parts of the body. So first statement is not correct. Then an exocrine gland releases substances. It makes directly into the fluid bathing the gland. No, it releases into tubes or duct. Exocrine gland releases into tubes or duct. So that makes both the statements as false. So answer will be option 2. Both statement 1 and 2 are false. Then next is again a match the following. Okay, mechanoreceptor. Mechanoreceptor means which will sense pressure. Okay, so 4 will be detects changes in pressure, position or acceleration. A4. Okay, you have it in 1 and 4. We will, so 2 and 3 are not correct. A4. Then nociceptor. So that will be detects tissue damage. B3. So that makes option 1 wrong. So 4 will be your right answer. Okay. Then we will see osmoreceptor. That is detect change in the water volume of a solution. And thermoreceptor temperature changes. That means respond to heat or cold. So that makes option 4 as the right answer. Then which of the following connective tissues has a matrix of collagen and elastin fibers in a rubbery ground substance? So rubbery ground substance is that which is containing your proteoglycans, glycosaminoglycans, etc. and this is found in cartilage. It has both collagen and elastin fibers, but the proportion of these collagen and elastin fibers vary with type of the cartilage. That is hyaline cartilage, if you take as an example, it has abundance of collagen and smaller amount of elastin. But if you take elastic cartilage, it has a matrix which is dominated by elastin and less of collagen. So the answer here is option 3, that is cartilage. Then next one, all of the following act as anaphylotoxins except. So what are these anaphylotoxins? They are small fragments released during enzymatic cleavage of the complement proteins. Now complement you might have studied, there are 3 pathways, that is classic, alternate and lectin pathway. And in all these 3 pathways you might have come across C3 convertase, C5 convertase, etc. And what do these convertases do? They will cleave C3 complement into C3A, C3B, and C5 convertase, it will cleave C5 to C5A, C5B. Okay. And this C3A and C5A, they will be entering into the bloodstream. The other C5B and C3B, they will be continuing with the pathway. So this C3A, C5A as well as C4A. C4A is also there. They are anaphylotoxins. Okay. And they are involved in inflammatory responses as well as allergic reactions. But C6A, it is not an anaphylotoxin. It is a part of the membrane attack complex and it is not an anaphylotoxin. So answer here is C6A. The mechanism by which B cells ensure that only one heavy chain and one light chain allele, okay, one only, is translated is referred to as allelic exclusion. So one allele is excluded. Okay. That is allelic exclusion, that only one heavy and one light chain allele is translated. That is called as allelic exclusion. So what is codominance? Codominance naturally found mainly with your genetics. That is, you have 2 different alleles of a gene and they both equally contribute to the phenotype. So the organism showing this codominance will have combined expression of the alleles. Okay, that is codominance. What is isotypic selection? What is isotype? B cells, they produce different antibodies. That is, it produces GAMED antibodies. So when the B cell switch from producing one type of antibody to another, that is from G, it is going to start the production of either A or E or D, then it is called as isotypic selection. What is idiotypic selection? Okay, before that, what is idiotype? It is the unique determinants on an antibody's antigen binding site. So the antibody is having an antigen binding site and this antigen binding site is having unique determinants, that is called as idiotype. And idiotypic selection means the immune system will select the particular specific antibody idiotypes. Okay, that is called as idiotypic selection. Then next one, given below are two statements regarding MHC. So MHC1 contains two different polypeptide chains, alpha chain, beta chain. MHC1 is present on all nucleated cells. Okay, before coming to the answer, we will see the structure of MHC1 and MHC2 also. Only then you will understand. Okay, now this is the class 1 MHC molecule. This is the peptide binding cleft, and this is the class 2 molecule. This is the peptide binding cleft. Now, if you see MHC1 and MHC2, MHC is having an alpha chain as well as beta 2 microglobulin. And MHC2, it is also having alpha chain and beta chain. Now in class 1 molecule, the heavy chain, this is the alpha chain, that is the heavy chain, and it has three domains, that is alpha 1, alpha 2, alpha 3. And there is a transmembrane domain for this alpha chain as well as a cytoplasmic tail. Now there is another one, that is the beta 2 microglobulin, and it is the light chain, and it covalently binds to the alpha 3 domain of the alpha chain. Okay. Now here in MHC2 you have got two different chains. One is alpha chain and the other is the beta chain. They both have transmembrane domains as well as cytoplasmic tails. Okay. Now class 1 MHC, it is like more of different polypeptide chains, not two different polypeptide chains. They are not two different polypeptide chains. Okay, it's a bit confusing, but I am telling you. But these class 2, they have two different polypeptide chains, alpha chain, beta chain. Here it is different polypeptide chain. Okay. And moreover, this class 2, these two chains, they are encoded within the MHC locus. So the genes present in MHC locus are involved in the synthesis of these two chains, whereas here only the alpha chain is synthesized by the MHC locus genes, whereas beta 2 microglobulin is synthesized somewhere else, not in MHC locus. Now come to the question. So MHC1 contains two different polypeptide chains, alpha chain and beta chain, is not correct. It has different polypeptide chains, that is not two different. Okay, it is different polypeptide chain, that is alpha chain and beta 2 microglobulin chain. Alpha chain and beta chain and two different polypeptide chain are present in MHC2, not 1. So statement 1 is not correct. Then MHC1 is present on all nucleated cells, that is correct. If you take the case of RBC, it is non-nucleated, it doesn't have MHC1. Whereas MHC2, it is found on mainly on the professional antigen presenting cells, that is macrophages, dendritic cells and B cells. Okay. So statement 1 is not correct, but statement 2 is correct. That makes option 3 as your answer. Then the next one. The function of 3', 5' exonuclease activity of DNA polymerase. Now they have told DNA polymerase and they have told 3', 5' exonuclease activity. So it is mainly with regard to the proofreading activity of DNA polymerase. So proofreading means it will remove any mismatches or incorrectly incorporated nucleotides from the newly synthesized DNA. Okay, that is what is meant by a proofreading activity. Now 3' 5' exonuclease activity means it removes the incorrectly incorporated nucleotide from the 3' end of the strand. See this picture, this is 5' to 3' DNA, and here I have shown dashed lines, which means that these are degraded removed nucleotides or incorrectly incorporated nucleotides which are degraded by the 3' to 5' exonuclease activity. So from 3' end it will start degrading the nucleotides. That in this strand also from 3' end it will start degrading. Whereas 5' 3' exonuclease activity means from 5' end it will start degrading, okay, the nucleotides. Now read the options. So removes the 5' end of the polynucleate strand. That itself is not correct. It should be removes the 3' end of the polynucleotide strand. So one is not correct. Then remove incorrect nucleotide from newly synthesized strand of DNA. Yes, that is correct. That is the proofreading activity of the DNA polymerase enzyme, and it is 3' 5' exonuclease activity. Then removes damaged nucleotide from the template strand, not from the template strand but from the newly synthesized strand. So 3 is also not correct. Remove nucleotide from the ends of the DNA molecule to ensure the generation of blunt ends. Again it is not correct. It doesn't generate any blunt ends. And then next one. Identify which of the following test is the most convincing to identify a gene as oncogene or tumor suppressor gene. So you have to identify whether you have to identify a oncogene as well as its tumor suppressor gene. So what you do, transgenic mice overexpressing the candidate oncogene. Okay, so you have got the oncogene O1 and its the tumor suppressor gene for this oncogene is T1. Okay, now you are overexpressing O1 in transgenic mice. Okay, the mice is going to get cancer. Okay, let it be. Then a knockout mice lacking the candidate tumor suppressor gene. So the mice is knocked out of T1. That is, there is no T1 production in that particular mice. So the mice is going to get cancer. So this transgenic mice and this knockout mice, they will show the same type of cancer. Okay. So you can conclude that yes, this particular oncogene is causing this type of cancer. So that you can conclude. Now since the knockout mice is lacking the T1, which is the tumor suppressor gene for this O1, you can conclude that yes, this T1 is absent in the knockout mice. It is developing cancer. So that means T1 is the candidate for this particular oncogene, or T1 is the tumor suppressor gene for this particular oncogene. So 1 will be correct. Okay. Now transgenic mice overexpressing the candidate tumor suppressor gene. So the mice is not going to develop any cancer. And knockout mice lacking the candidate oncogene. So it is having only the tumor suppressor gene and it is not going to develop cancer. So both mice are not going to develop cancer, and you cannot tell that okay, this particular gene is causing cancer. That conclusion you cannot make out from this experiment. So 2 is incorrect. Then transgenic mice that overexpresses the candidate oncogene and tumor suppressor gene, no cancer development at all. So you cannot conclude anything telling that this gene is the one which is oncogenic and the other one is the tumor suppressor gene for this particular oncogene. You cannot tell. So 3 is also not correct. Knockout mice that lacks the candidate oncogene and tumor suppressor gene, what is the fun? Nothing you can make out. there is no oncogene, no tumor suppressor gene, nothing is going to happen to the mice. So your study will be a failure in case of option 4. So 1 is the right answer. Then next one. A study is done on a mammalian cell line that has a doubling time of 24 hours. These cells are synchronized in G1 and then labeled for 2 days with BRDU, that is a base analog of thymidine. So it is a mutant. At the end of the labeling period, chromosomal DNA is isolated from the cell and its density analyzed by equilibrium centrifugation in cesium chloride gradient. It is nothing but your cesium chloride density gradient centrifugation. But they have told it in a roundabout way to confuse you. That is what they meant by the statement. Density analyzed by equilibrium centrifugation in cesium chloride gradients. So density of the DNA, it is analyzed by equilibrium centrifugation in cesium chloride gradients. That is nothing but cesium chloride density gradient centrifugation that you did when you studied when you learnt semi-conservative method of DNA replication. So which of the following patterns can be expected to be seen? Okay. Now this mammalian cell, it is having a doubling time of 24 hours. So 1 day, 1 division, second day, second division. These cells are synchronized in G1. Okay. Now all the cells that you are going to study, they are in their G1 phase. So you are conducting a race for cell division, means all will align at the starting point, will be in the G1 phase. Now from there they will start moving to other stages of cell division. Okay, from G1 it will go to S, then to G2, then to M. So that is the meaning of G1, synchronized in G1, and then labeled for 2 days with BRDU. That means 2 generations they have studied, because 1 generation is 24-hour cycle. So 2 days means 48 hours. So 1 day, 1 division, second day, second division by adding BRDU. So and what they did, they isolated the chromosomal DNA and conducted cesium chloride density gradient centrifugation. What is the output? So suppose you have a gene sequence GAC CTG, okay, that is in your G1. Now what you did, you are allowing it to grow. So it has gone to the S phase. S phase, you are adding that BRDU. So this is the division, it is semi-conservative. So the 2 strands separate, and upon replication, instead of thymine, BRDU will be incorporated in 1 strand. The other strand, it will be adenine incorporation, because thymine is there already. So it will be adenine and it will go for normal round of replication. It is not mutated, but here it is mutated. They have asked for this one, the mutated one replication. Here it will be always same, that is it will be 100% heavy chain. Okay, this one, but they have asked for the at the end of labeling period. That means after labeling, what is the outcome? So again, this is after 24 hours. So this is the first replication, and this will be your second replication. Again the strands have separated. This will be full of your parental, heavy chain, and the BRDU labeled one will be your light chains. So here it is the BRDU labeled strand, and it will, upon semi-conservative replication, it can incorporate either, it is a thymine, so it can incorporate either adenine or a guanine. So I am not going into details regarding that. They have not asked about whether it is adenine or guanine that is incorporated. They have asked only regarding the density of the chain. So BRDU chain, it is the light chain. The other one will be your heavy chain without the BRDU. So you will get 50% will be HH and 50% will be HL. So that makes the answer as option 3. Now, identify the correct order in which extracellular signals are transmitted. So here it is the adenylate cyclase, CAMP, protein kinase. They are all involved with the G protein coupled receptor activation. So extracellular signal is coming, the receptor is bound with that particular protein or hormone or whatever. This is your G coupled protein receptor, and this is the G protein having alpha, beta, gamma subunit. The G protein gets activated, GDP is removed, GTP is exchanged for GDP, and it will result in the activation of adenyl cyclase. Adenyl cyclase, it will activate CAMP, and CAMP will activate protein kinase A, and protein kinase A will in turn result in phosphorylation or activation of the downstream proteins or other cellular materials. So here the sequence they have asked. So first will be adenyl cyclase, then CAMP and protein kinase A. That means ABC. So the answer will be option 1. Then which of the following is not a function of lysosome? Okay, lysosome, suicidal bag. They will digest everything, including your own cells if they are damaged. So autophagy is there, then outside substances will be engulfed, and they are suicidal bags. They have acid hydrolysis. So the not a function they have asked. So it will be power generating units. So that means power generating units are nothing but your mitochondria. Okay. So that's it now. Thank you for watching my channel. Please do like, share and subscribe. If you have any doubts, you can write it in the comment section. And if you want still previous year question papers of ICMR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, see for ICMR, and you will find them there. Thank you.